I want to sort of add my thanks to Tony and all the leadership for putting this together. I'm not really an excitable type, but I'm very excited about the Night Initiative generally and about our project in particular, so thanks for funding it. So I'll just quickly introduce the team. That's me. I'm a behavioral neurologist, and my group's interested in genetics of Alzheimer's disease, particularly leveraging extreme phenotypes, which I'll talk about in a bit. Dr. Raventos is our PI at the University of Costa Rica. She's a medical doctor, a physician scientist in the Department of Genetics there, and vice president of the National Academy of Sciences in Costa Rica. Ewan Ashley is in internal medicine, and he's really helped our group tremendously with long-read sequencing. David Reckhoff is in epidemiology and population health. I think I saw him. Yes. He's interested in social and economic determinants of health and also epigenomics. And then Hua Tang from genetics is going to help us think about the complex admixed ancestry in this population. And then I wanted to recognize some of our junior colleagues, Ted Wilson, Patricia Moran-Lasada, and Christina Young, who are going to be helping with Alzheimer's biomarkers, plasma proteomics, and neuropsychological profiling, respectively. All right, so my group's really interested in aging and the effect of ApoE4 on Alzheimer's disease. This is a really beautiful study actually from Henna's colleagues in Rotterdam. This is the population or the community-based Rotterdam study. And this just shows you the cumulative incidence of Alzheimer's disease with age and as a function of the different common ApoE genotypes. And you can see, I guess you can't see, I don't have a photo, but the purple bars there are E4 homozygotes. So carrying 2 copies of E4 is a gigantic risk factor for Alzheimer's disease that increases with age. So our group is interested, for example, in finding older E4 homozygotes who aren't sick and looking at their genetics. And the flip side of that, people that are E3 homozygotes but have Alzheimer's disease, for example, thanks Tony, in their 50s. And that's what we think of as extreme phenotypes, and we hope that even if our sample sizes are small, that sort of extreme phenotype will enrich our capacity to find rare protective variants or rare causal variants related to Alzheimer's disease. I should point out this is actually a little rosier than it might look. This is just Alzheimer's disease cumulative incidence. The slide below it was all-type dementia, and this goes up to about 70%. So it's tough if you're an E4 homozygote and you live to 85 or 90 to escape all types of dementia, but Alzheimer's is the big player there. Okay, and our group has been interested also more recently in sort of the ancestral background and how that affects risk related to ApoE4. This is from a nice review from Michael Beloy in our group, looking at the frequency of the ApoE4 allele across the world. And what you can see right off the bat is that it's highest in sub-Saharan Africa. We think this is the ancestral variant, the sort of out-of-Africa variant. And it's quite low and uncommon, for example, in parts of East Asia, so China and Japan. And what's really interesting is there's sort of an inverse correlation between the frequency of the E4 allele in a given ancestral background and its Alzheimer's associated risk. And that's shown here. So most of our work still today, at least in the US, most of what we understand about ApoE and Alzheimer's disease is in European ancestry cohorts. And if we think about the odds ratio for Alzheimer's disease, if you have 1 copy of E4, you bump your risk up about 2-fold compared to E33. If you have 2 copies, you bump up your risk about 12 or 14-fold. It's a really powerful risk factor on a European ancestral background. The risk of carrying 2 E4 alleles on an African ancestry background is considerably less, more like 5x, so about half as potent an effect on an African ancestry background. And by contrast, on an East Asian, in this case Japanese background, the odds ratio jumps up to about 25 or 30. So there are really massive differences in the sort of E4 attributable risk of Alzheimer's disease across these ancestral backgrounds. And so our group has leveraged this in some ways to think about variants that either increase or decrease risk for Alzheimer's disease on or near ApoE. And I'll kind of run through this quickly. First of all, we're mostly interested in local effects, but we've definitely found distant effects. So for example, this Clotho VS variant is protective in E4 carriers. That's not that close to ApoE. But most of what we looked at are variants either on or near ApoE. So these two are rare variants that are seen in Europeans. They're both protective that we published recently. And then hot off the presses, this is our first foray now that the NIH has been really supportive of generating cohorts of diverse ancestral backgrounds. The sample sizes are large enough that we were able to find this risk-increasing variant on ApoE, R145C, which is actually quite common in African Americans and very rare in European Americans. So we're beginning to sort of piece together some notions about how individuals from different ancestral backgrounds might protect themselves against the Alzheimer's effects of ApoE. Okay, so for the Night Initiative project, this is going to involve clinical and cognitive phenotyping of a large cohort of very long-lived Costa Ricans. There are two clusters, actually, of centenarians in Costa Rica in these two different regions, and I emphasize the true here because Costa Rica, among its many fantastic attributes, is very good about record keeping. It has a fantastic National Health Service. So when somebody in Costa Rica says they're 100, they have a card that says that they're actually 100, and you can count on it. It's a very admixed population, which is important for the sort of work we want to do. So about a third European, Amerindian, and African ancestry, many times in the same individual. And interestingly, E4 frequency appears relatively high in these 90-plus-year-olds, so 13 or 15%, which we don't see here. So the plan will be to do 30x coverage, long-read sequencing with Oxford Nanopore technology, with you and Ashley's help. And we'll be looking at single nucleotide variants, but also structural variants, and a quick plug for long-read sequencing. This is the same subject. This is a portion of TMEM106B, a gene in lysosomes, actually, that Malta might be interested in. This is the short-read sequencing from this particular individual, and this is the long-read sequencing, which picks up this massive deletion, about 300 base pairs that was totally lost to the short-read sequencing. So we're excited about the long-read sequencing. It'll help us phase variants with E4. We can also get a methylation readout from this long-read sequencing approach. Ted's going to help us with plasma biomarkers, and then Patricia from Tony's group is going to help us with a broader look at plasma proteomics. And at the end of the 3 years, we expect to have done this multiomic profiling on 560 90-plus-year-olds, and as a comparison group, about 150 younger controls, with the hope of characterizing these variants, and ideally, not our group, but working with others here and elsewhere, identifying druggable pathways. And then ideally also establishing a longer-term collaboration with Dr. Raventos and her team in Costa Rica to think about other age-related phenotypes, of which, of course, there are many. And I'll thank my lab and collaborators here, and 30 seconds late. Thanks.
We have time for one question.
Okay, I'm wondering whether the differences that you see in ancestry, if any confounds have really been completely excluded. So for example, if there is a bad gene like ApoE4 and there is a population that has particular susceptibility to its atherosclerosis side effects, you could imagine that there is a survivor effect where they get taken out by the atherosclerosis effects, and now it looks like there is less of an impact because the ones that are really super susceptible, they're all gone by the time you look for dementia, right? I mean, so I was just curious whether that had been considered.
Right. Yeah, thanks, Leonard. That's a great question. And the short answer is not really. I think we're going to have some ability to think about these things because in addition to the genetics and the cognitive and clinical phenotyping, they have a nice sort of record of social exposures, education, all these other issues. But you're right, E4, among the many things it does, also increases cardiovascular risk, obviously. And so for all of these cohorts, I think there is a bit of a survivor effect, which is tough to model, but important, definitely. Thanks.